Friends, in the last lecture itself, we have started with the discussion of chapter number 8 and it is titled as Respiration and Circulation. And in that, the first thing we have studied, it was nothing but the basic information about the respiration, and in the second lecture, we have discussed the respiratory system of human being. And then today we are going to proceed on to the next topic. Friends, cells require a constant supply of different substances like oxygen and nutrients for performing life activities. Similarly, there is a need of constant removal of waste materials produced during metabolism like CO2 and nitrogenous waste material. Now this function is served in higher animals by a highly specialized transport system called circulatory system or cardiovascular system. Now important terminology, it includes angiology. Now this angiology is defined as study of blood vascular system, whereas cardiology is the branch of biology which deals with the study of heart and related functions, whereas hematology is the branch of biology which deals with the study of blood and the variety of compounds that are included in the blood. Then we'll study functions of circulatory system. The first function is nutrients and waste product which are produced during the metabolism, they are transported through circulatory system. Secondly, respiratory gases like oxygen and carbon dioxide, they are also transported through circulatory system. Now this circulatory system, it carries metabolic intermediates which are very important for survival of the organism. The hormones which are produced by endocrine system, they are also transported to the target tissue with the help of circulatory system. Now whatever nutrients are available, they are uniformly distributed like water, the protons and chemical substances along with the body heat. And lastly, circulatory system plays important role in the defense system of the organism against various types of diseases. Now in this particular diagram, we will see single circulation like in fishes. Now we know in case of fishes, a two-chambered heart is there, that is one auricle and one ventricle. That means the blood, deoxygenated blood, it enters into the heart and then into the auricles, then to the ventricles, and from the ventricle the blood is pumped out. So the blood enters into the heart in deoxygenated condition and it leaves the heart in same deoxygenated situation, and that's why it is referred as single circulation. Now single circulation, it is found in lower organisms like fishes where heart is two-chambered. Now blood passes through once through the heart and it is obviously less efficient system. Oxygenated and deoxygenated blood, they are mixed and circulated. As a result of that, the animals become lethargic as they have got less supply of oxygen to the different organs. Now second mechanism is double circulation. Now here you can see four-chambered hearts are there, and when the four-chamber hearts are there, the right side of the heart, blue colored structure in the diagram, it is dealing with the deoxygenated blood, whereas this red colored one, it is dealing with the oxygenated blood. So complete separate circuits are available for deoxygenated and oxygenated blood. Now this double circulation, it is generally found in higher animals like amphibians, reptiles, birds and even the mammals. Blood passes twice through the heart and it is the most efficient system as rapid transportation is available. Oxygenated and deoxygenated blood, they are separately circulated, so there is no mixing. As the animals and the organs are getting oxygenated blood, obviously they show more activity, or they are, we can say, having sufficient supply of oxygen to the different organs. Then we'll go for the first component of the circulatory system, and obviously it is nothing but the blood. Now blood, it is highly specialized liquid connective tissue. Remember, it is the only tissue which is in liquid state in the kingdom Animalia. Now it constitutes 8% of the total body weight and 30 to 35% of the extracellular fluid. Now in an adult healthy person, blood volume, it measures about 4.5 to 5.5 liters. Now the viscous nature of blood, it is due to presence of serum albumin, the protein which is present in the plasma as well as blood. Now blood is red in color due to presence of respiratory pigment, that is called as hemoglobin, and it is slightly alkaline with pH 7.35 to 7.45. Now that pH of blood in artery is more than in the veins, and actually blood is containing a buffer that does not allow the change in the pH of blood, and buffer of the blood is sodium bicarbonate. Then we'll see composition of blood. The liquid connective tissue blood, it consists of two components. The fluid part, that is called as fluid plasma, and this plasma contains suspended cells, that is blood corpuscles. So in that manner, the blood can be compared to a colloidal substance which contain insoluble components and soluble components together. The first component is plasma. Now it is the ground substance, colorless alkaline fluid, and that form 55% of the total blood. About 90% of it is composed of water, and only 7% it consists of plasma proteins. Now these plasma proteins include serum albumin, serum globulin, heparin, serum fibrinogen and prothrombin. Now serum albumin, it provides osmotic pressure in plasma, whereas globulin is a protein antibody that provides immunity to the organism. Protein heparin is acting as anticoagulant, and serum fibrinogen and prothrombin are important for blood clotting. Now these plasma proteins are efficient buffers, and they help to maintain the pH of blood. Remaining 3% are nutrients like glucose, amino acid, fats, inorganic substances like chlorides, bicarbonates, sodium, potassium, calcium, magnesium, ions, organic substances like enzymes, hormones, respiratory gases like oxygen, carbon dioxide. Additionally, they contain excretory material like urea, uric acid, creatinine, etc. Now, plasma without fibrinogen. When fibrinogen, the blood clotting protein, is removed from the plasma, it is termed as a serum. Second component of blood, it is called as blood corpuscles. Now blood corpuscles, they form 45% of the total blood suspended in the plasma. The process by which blood cells are formed, it is called as hematopoiesis or hemopoiesis. Now blood corpuscles in higher organisms, they belong to three categories: the RBCs, red blood corpuscles, WBCs, white blood corpuscles, and platelets. The first blood cell, they are called as RBC, red blood corpuscles or erythrocyte. Now these are small, circular, biconcave, and they have a diameter of around 7.7 micrometers. They are thin in center and thicker towards the periphery. So they are called as biconcave. These are non-motile, non-nucleated in all mammals except llamas and camels. Absence of nucleus help to accommodate maximum amount of hemoglobin. RBCs, they contain red pigment, red colored pigment, that is called as hemoglobin. Now hemoglobin is an conjugated protein formed by protein part called as globin and a non-protein prosthetic part called as heme. Now hemoglobin combines with oxygen to form oxyhemoglobin, and the enzyme carbonic anhydrase is present in RBCs, which convert the water molecules and CO2 to form H2CO3, which is later on combined with the either potassium or sodium to form KHCO3 or NaHCO3. In the form of these carbonates of sodium and potassium, RBCs transport 70% of CO2 to the lungs. Hemoglobin can carry 23% of CO2 in the form of carbaminohemoglobin. Normal range of hemoglobin in human being, it is 14 to 17 grams per 100 ml, and in women it is little lesser, 13 to 15 grams per 100 ml. A normal healthy man has 5.1 to 5.8 million RBCs per cubic millimeter, and normal healthy woman has 4.3 to 5.2 million per cubic millimeters. Now RBCs has a definite lifespan of about 120 days, and then they are destroyed in the spleen. Now that's why the spleen is called as graveyard of RBCs, or the Kupffer cells of the liver are responsible for destruction of worn out RBCs. Iron of the heme group is extracted and stored in the liver as ferritin, and then it is reused later for production of new RBCs. Reminder that heme molecule is broken down into bile pigments such as bilirubin and biliverdin that later on excreted in the gut by the way of bile. RBCs are formed in liver and spleen in fetus, while in adult they are formed in the red bone marrow at the rate of 1 million per second. Now each day around 20,000 millions are produced and destroyed. Second group, it is the WBCs, white blood corpuscles or leukocytes. They are the most active motile constituent of blood as well as lymph. They are colorless as devoid of hemoglobin, and these are irregular amoeboid cells than the RBCs. Total leukocyte count, that is TLC, is about 5,000 to 11,000 per cubic millimeter. In acute infections like pneumonia, the total number of WBCs, they may increase up to 20,000 or more per cubic millimeter of the blood. During pregnancy and in newborn, the number of WBCs increases about 10,000 per cubic millimeter, and that is called as leukocytosis. If the number falls below 4,000 per cubic millimeter, then such condition is called as leukopenia. Now leukemia is a pathological increase in the number of leukocytes and many a time referred as blood cancer. The ratio of WBC to RBC for a normal healthy person is 1:600. WBCs are nucleated and capable of amoeboid movement. Now this movement of WBCs within the bloodstream, it is called as diapedesis. Now these WBCs are produced in spleen, yellow bone marrow, and when worn out, they are disintegrated in bloodstream and engulfed by phagocytic cells that are called as phagocytes. Lifespan of a WBC is very short, around 10 to 13 days. Now based on the nature of their cytoplasm, shape of nuclei and staining property, leukocytes are of two types. The first one granulocyte and the second one agranulocytes. Now if you see the granulocytes, they have granules in the cytoplasm and polymorphic nucleus. They form around 72% of the total WBCs. Now they are divided into three types on the basis of their staining properties: acidophil, basophil and neutrophils. Now, as far as acidophils are concerned, they take acidic stain like eosin, hence called as eosinophils. These are having bilobed nucleus. As you can see in the diagram, these are having bilobed nucleus. It is a characteristic feature. Now they have diameter of about 10 to 14 microns and lifespan of about 14 hours. The main function of acidophil is to produce antihistamine, which destroy toxic substances produced by antigen antibody complexes. The number of acidophils present in the blood serum is under control of hormone, which is secreted from the adrenal cortex. The second category is basophils. Now basophils, they take up stain which are basic in nature like methylene blue, and the nucleus is generally 2 to 3 lobed. You can see this 2 to 3 lobe nucleus, and often it is S-shaped spiral. Now these are smallest WBC and about 8 to 10 microns in diameter. Lifespan of basophil is 8 to 12 hours Basophils produce histamine, a chemical compound which is released during the allergic condition. They also release heparin in the blood, which act as anticoagulant. The third category is neutrophils. Now these neutrophils, they do not take any basic or acidic stain. Nucleus is twisted, multinucleated, multilobed, 3 to 5 lobes are found. They measure about 10 to 12 micrometers in diameter and mainly phagocytic in function. They attack and engulf the materials which are useless for the body. Their granules contain hydrolytic enzyme capable of digesting the bacteria as well as other pathogens. Second category of WBCs, they are called as agranulocytes. They have clear cytoplasm without any granules and nucleus is not lobe. They form around 28% of the total WBCs of the cell. They are divided into two types. The first one monocytes and the second one they are called as lymphocytes. Now the first category is monocyte. Now these are large cells with kidney shaped nucleus. Here you can see the kidney shaped nucleus is present characteristically and they have got large amount of cytoplasm. They constitute 40% of total WBC and measure about 16 to 20 microns in diameter. They are highly mobile, phagocytic in function and they spend 30 to 40 hours in blood and then they enter into the tissues where they become macrophages, which are phagocytic in function. Second category is lymphocytes. Now lymphocytes, these are smaller cells with large spherical shaped nucleus. Here you can see the spherical nucleus is there and very little cytoplasm. They constitute around 24% of the total WBC. Measure 8 to 12 millimeters in diameter. Their lifespan ranges from months to years and they are found in the thymus gland, lymphoid tissue and then they are functioning in the blood. The main function of lymphocyte is to produce antibodies which are helpful for the immune system. The third type of blood corpuscle, they are called as thrombocytes or platelets. Now these are irregular cytoplasmic masses. Now you can see these are the irregular cytoplasmic masses without nucleus. They are developed from erythrocyte as well as leukocyte. Variable size and size ranges from 2 to 4 microns in diameter, quite small. Platelets which are larger, they are called as megakaryones. The formation of platelets is regulated by a hormone that is called as thrombopoietin and their formation is known as thrombopoiesis. Lifespan of thrombocyte is very short, 5 to 9 days and they constitute around 1,50,000 to 4,50,000 platelets per cubic millimeters of blood. They play important role, vital role in clotting of blood during wound and they release the substance called as thrombokinase. Now platelet release serotonin which constrict the blood vessel and thereby reducing capillary bleeding. A decrease in the number of circulating platelet is called as thrombopenia and such person have tendency to bleed from capillaries all over the body. Friends, then we'll see the functions of blood. The first function is obviously transportation of gases, nutrients, minerals, hormones and metabolic waste. Secondly, they provide immune power due to lymphocyte. Thirdly, they are responsible for blood coagulation due to thrombocytes. They maintain the body temperature by dissipation of heat. They maintain the water balance of the body, salt and water balance of the body. Regulation of pH is the next function. They also provide storage function for nutrients as well as the waste material and very importantly they are helpful for removal of dead cells, debris from the body. So friends, today we have studied the first component of the circulatory system and that is in the form of blood. I hope you are understanding it very well and it will be helpful for your studies. Thank you very much.